Viral infections are part of our everyday life and constitute a huge threat to human health. But because we cannot see viruses, often we do not really understand how they spread within the body and how our immune defenses counteract their propagation. Recently, the zebrafish has started to make waves in this field. There are many genetic tools available in zebrafish, but its unique experimental advantage is imaging at early stages. It is transparent, small, and easy to anesthetize. In zebrafish larvae, with a virus containing a fluorescence gene, you can follow infection in real time and see every infected cell in the whole organism.
The first example of such a real-time observation of a viral infection has been made with the human pathogen, chikungunya virus. Chikungunya virus is a mosquito-transmitted virus that was relatively ignored until 2005, when it caused a large outbreak in the Indian Ocean. As of today, it continues to invade zones where it was absent previously. It is a serious health concern, especially for newborns, which can get killed or crippled as they develop encephalitis.
In this movie, this zebrafish larva has been infected with a modified chikungunya virus that makes infected cells to express GFP. GFP-positive cells appear in various places. Strikingly, after the first day, and despite an enormous production of new infectious variants in the larva, almost new infected cells will appear. The infection is stopped in its tracks by the potent endogenous interferon response. The number of infected cells will steadily decline. Apoptotic death of infected cells was observed throughout the animal, as you can see here. An unexpected finding was that the death rate of infected cells was strongly organ-dependent. Infected hepatocytes died 2 times faster than cells elsewhere in the body. In contrast, infected neurons persisted for as long as they could be observed and became the only virus reservoir after a few days.
This virus induced a strong interferon response in larvae, just as in mammals. This zebrafish transgene labels with red fluorescent cells that express the highly inducible type 1 interferon, so the host response can be imaged in real time along the infection. Liver cells were found to make interferon along a population of mobile leukocytes patrolling everywhere except in the central nervous system. These were neutrophils. Genetic-based depletion experiments showed that these neutrophils were critical for the larvae to control the virus infection.
Along virus dissemination tactics and host countermeasures, another question that could be addressed efficiently in the zebrafish is that of tolerance versus resistance. While resistance responses aim at reducing the pathogen burden, tolerance ones allow the host to remain healthy even in the presence of high microbial loads. To analyze tolerance, one has to be able to measure easily the microbial load and the health status of the host. Assessing health status of zebrafish larvae may sound challenging, but is actually easier than in mice. Combining basic behavior with internal parameters such as blood flow or heartbeat, an health score was built and confronted to the measured viral load. In the case of chikungunya, the limited data available suggests that the zebrafish iNOS gene may contribute to tolerance more than to resistance.
In the near future, we will undoubtedly visualize the interactions of zebrafish larvae with other viruses using diverse spreading strategies. Combined with the possibility of performing genetic or chemical screens in 96 well plates, we predict that the zebrafish will become a major piece to solve the puzzle of virus-host interactions in the coming years.